He kōna e purangi tēnei nā te reo irirangi o Aotearoa.
Professor James Bell and Kea Witting have just finished a dive.
Yeah? Yeah, it looks very pretty down there with all the little sponges.
They're handing equipment up to PhD researcher Gabi Wood before they haul themselves into the little black and orange rigid-hulled inflatable boat. They've been down for 39 minutes. 39 cold minutes, says James.
How cold was it?
14.
It's the same as in Wellington right now.
Minimum 13.
There's one little dip into 30.
Yeah.
It's like
Winter
13.
Wellington weather, Irish summer weather. Yeah.
Ready for a cup of tea?
Cup of tea time.
Kia ora, nau mai haramai ki te ao hurahanga. Hello and welcome to Our Changing World, ko klekan kanan tēnei. Wellington winter equal to Irish summer? Sounds about right. Last June, while the continent of Europe was baking in a heat wave, I took a detour from a family trip to join a team of Wellington-based researchers who were at, of all places, a cold sea inlet in West Cork in Ireland.
Yeah, probably as far away from Victoria University of Wellington as you could possibly get, I reckon.
Today on Our Changing World, we bring you an underwater mystery. One spanning decades and connecting distant continents. A story steeped in scientific history, filled with as yet unanswered questions, but with a hopeful ending. It is the mystery of the disappearing sponges of Lough Hyne. Lough is the Irish language word for a lake or sea inlet. And there are many places to start this tale of Lough Hyne. Maybe 4,000 years ago, when the sea level rose enough to turn this freshwater lake into a saltwater basin filled by the Atlantic. It could begin with the first recorded biological study of Lough Hyne, a note of the purple sea urchin in 1886. Or with the first hut assembled in 1928 as an on-site research lab. We could leap into the 1950s, a time of multiple sturdier labs and lots of scientific interest. And new species were regularly being discovered in the loch, alongside key marine ecology findings about how species interact. Or we could start in 1981, when Lough Hyne was designated Europe's first statutory marine nature reserve. But we're going to focus on the New Zealand connection and start instead with when James Bell first came to Lough Hyne as an undergraduate student in an undisclosed year, sometime in the 1990s.
Back in a little while ago, with my supervisor then, Professor John Turner from University of Wales, Bangor. So I spent 4 weeks here, absolutely loved it. We camped out for the 4 weeks, we dived every day. And then there was an opportunity after my degree to come back to Lough Hyne and do a PhD. So I did my PhD at University College Cork, and I finished that in 2001. My PhD was largely focused on the unusual sponge communities that you find inside Lough Hyne. So Lough Hyne's a really unusual place. It's got a really wide range of habitats within a really small area. And one of the things it's particularly well known for is these subtidal cliffs that are absolutely coated in sponges. At the time when I did my PhD, I used to think they were super unusual and super rare, and over time we found that they're maybe not as rare as we once thought, but they're quite characteristic of the sorts of communities that we normally find in deeper water.
James took up a job at Victoria University of Wellington in 2006. And today, much of his research work involves diving or using remotely operated underwater vehicles to survey and understand sponge communities in New Zealand and at research sites in Honduras and Indonesia. It would be 15 years after his PhD had finished that he would return to Lough Hyne. A 2016 International Sponge Conference in Galway brought him back to the Emerald Isle.
And as a treat to all of my PhD students who came with me, I told them about this amazing place I did my PhD, where there are all these cool sponges, and I brought them all down to Lough Hyne. And I think most of them probably thought I'd made up all of the data in my PhD because most of the sponges on a lot of the subtidal cliffs had actually disappeared. That sort of set the scene for me coming back to Lough Hyne to, I guess, try and figure out to start with what had happened and quantify what happened. More recently, that's kind of then focused on trying to understand if there's any recovery of those sponges over time. And then this year we've started a new project funded by the National Parks and Wildlife Service to try and restore some of the sponge assemblages in Lough Hyne.
The National Parks and Wildlife Service is the Irish equivalent of the Department of Conservation. They've been funding James and his UCC, or University College Cork, collaborator, Professor Rob McCallan, to address those three questions. Why did the sponges disappear? Are they starting to recover? And can they be helped to return?
Go. You want me to put that into your...
Gabi Wood and Kea Witting are getting ready just outside of the Renouf Lab at the edge of the loch. It's a small white building, a little shed beside it, surrounded by bracken and ferns. It's maintained by UCC for use by researchers. Inside is a lab space, a workroom, a small kitchen and food storage area. Off to the side is a kit room and a toilet. And upstairs are 2 rooms, each with 3 single beds. This is where Gabi and Kea have been staying for several weeks now. They're about to head out for a dive, so they perform checks on equipment and hang what they need off hooks on their dive suits.
I got both cameras, my knife, my torch. One of my dive instructors always used to say, if you don't go down diving like a Christmas tree with all the stuff on it, you're not doing it right.
The lab is boat-only access, and the one that picked me up was timed to coincide with the delivery of liquid nitrogen from UCC. It's needed for the samples they'll be collecting on this dive at a site called Whirlpool Cliffs. But it's not looking whirlpooly right now because of the tide, which is part of what makes Lough Hyne so special. Here's James.
It's small. It's only about 800 metres long, about 600 metres wide. It's a fully marine sea loch. There are a couple of very small streams that feed into it, so there's a little bit of fresh water, but it's largely driven by the processes out on the open Atlantic coast. So it is tidal, but it has kind of a really unusual tidal flow regime. There's a what's called the rapids, which is like a narrow channel which connects Lough Hyne out to the Atlantic Ocean. It's only about 25 metres wide, and at high tide it's only about 3 metres deep. So it's very shallow compared to the loch itself, which is over 50 metres deep in its deepest part. And there's a rocky sill that runs across this channel, and the water, as the tide comes up, the tide has to reach the level of that sill before it can flow into and pour inside Lough Hyne. So over a normal 12-hour tidal cycle, what that means is the tide flows into Lough Hyne for about 4 hours and out for about 8 hours. And what that results in is, basically, it's very calm in the loch at the moment. There's no water movement. So when the tide's going out, the water gets skimmed off the surface out to the Atlantic. When the tide comes in, it builds up and builds up and builds up. It reaches the level of that sill, and then it pours in really fast. So you get these really, really fast water currents just in this area over here, where the guys are diving in Whirlpool Cliff. The water then gets deflected off Whirlpool Cliff, heads across the loch, and as it does, it slows down really quickly. And as the water slows down, the sediment settles out of suspension, and you get this really strong gradient of flow and sediment as you go from east to west across the loch. So you get lots of different environments within this really small space, and that's what supports the huge biodiversity that you find inside Lough Hyne.
The rapids were endlessly fascinating to me. It's a short walk from the lab, and I managed to get there as the tide made the switch from coming in to dropping. I watched jellyfish being swept along in this lazy river before everything slowed down, then stopped. The big fronds of kelp that had been bent one way lifted up to flap vertically for a second or two before bending the other, the same jellyfish passing by, but now heading the other direction, back out to sea. Now, with the lower tide, Whirlpool Cliffs is calm and diveable. After an okay hand signal, Gabi and Kea sink below and we sit and wait. Gabi is investigating what the sponges are eating and whether there's enough of it. To do this, she uses needles attached to tubes to slowly collect water from inside and outside the sponge to identify what the sponge is filtering out to snack on. They're back up after about 30 minutes, and it's a super quick spin back to the lab where Gabi starts to process the samples.
It went very well. We were able to get a good drip, and it was about 15 minutes each. And we got it at the same time.
So 2 different species?
Yeah, 2 different species. One is Cleona. It's a big yellow sponge, quite gorgeous. It looks kind of like cheese in a way. And then the other one is Cetethia. It looks much more like a giant golf ball, like a giant orange golf ball.
And when you say giant, what kind of size are we talking?
Like kind of like your fist. If you take your fist and pretend that's a sponge, that would be the Tethia.
And you're labelling some tubes. What
Yes.
happens next?
What's going to happen next is we're going to take the liquid that we collected and we're going to put it into these tubes. They're called cryovials, and they are able to withstand negative 80 degrees Celsius temperatures. So really, really cold, and they won't break. So we'll put it in there so that we can then analyze these water samples for all the different types of plankton and organisms in the water later on. And it preserves the organisms quite well in liquid nitrogen.
So samples into tubes into the liquid nitrogen.
Yep,
And then they'll be frozen until you can get to them
that's right.
back in
Yeah.
Wellington.
Yes, still making a big trip to the southern hemisphere. Yes.
So what could be behind the sponge's mysterious disappearance? The team would be surprised if it came down to lack of food, but they need to check it out to be sure. So far, they've tested quite a few hypotheses based around your usual suspects.
Changes to sedimentation regimes in the loch, changes in the amount of nutrients coming into the loch, climate change impacts, particularly heat waves or also cold spells.
But these don't seem to be the problem. It was Gabi who did the nutrient work. She looked at the impact of increasing amounts of nitrogen-containing fertilizer, both with and without warming temperatures. She ran those experiments on some sponges in Lough Hyne and also some species found in Wellington. And both sets of sponges seem to be very resilient to nutrient changes. There's another thing that happens in the summer in Lough Hyne, where you get the water separating into layers based on the temperature, called a thermocline. And the deep layers can be full of bacteria that use up all the oxygen down there.
And one of our hypotheses is that maybe that deeper layer has somehow become inverted very quickly and then bathed all of those cliffs inside Lough Hyne with that oxygen-poor water. And that's what's killed off all the sponges. But we've done a whole bunch of cool experiments on sponges that took a lot of time. And we found actually if you take most of a sponge's oxygen away, somehow it still survives. So a whole different branch of research.
Right, so in trying to figure out this question of what caused the decline of sponges here, you've discovered something
Yeah, we
that
discovered
people
something
didn't
really cool.
know.
That's right, we've discovered something. And what's super interesting about that is that, so we did a bunch of experiments here at the loch where we took away the sponge's oxygen and saw what happened to them, which is pretty much nothing. And then we repeated the same experiments on sponges from New Zealand. And we found, again, if you take away all of their oxygen, they don't particularly care either. And we think that's probably related to their really long evolutionary history and the relationships they have with microbes that live in their tissues. But yes, a whole different story to Lough Hyne, but these kind of interesting things that you find out along the way of other projects. So we know it's not oxygen. Our kind of last thing that we think it might be is potentially the presence of hydrogen sulfide in the deeper water layer. So as well as that deeper water having low oxygen, we also think that there might be high levels of hydrogen sulfide, which is toxic to most organisms. And perhaps that caused the decline in sponges again through some kind of weird breakdown or inversion of this deeper layer. But unfortunately, we plan to do those experiments this summer, but the thermocline hasn't formed enough yet for there to be hydrogen sulfide in the water. So those experiments will probably get carried forward to another year. But we've kind of done experiments and observations on lots of things, and we've struggled to really narrow down exactly what that causes, I guess, which is very, very frustrating. I have to say, very frustrating.
It leaves them in a tricky situation too. Because if you don't know what the problem was, how can you be sure that it was a short-term issue that's now resolved? The positive news is that there does seem to be recovery of the sponges in some of the cliff areas of the loch. That's what Kea will be focusing on for her research.
Part of my PhD project is to look at all the different sites in the loch, and then also over the period of time that we've been taking photo quadrats at all the different sites for twice a year. I look at all those photos and I assess the recovery of the sponges that we lost in the mass mortality event between 2010 and 2015. So my time series starts in 2018 and I have photos of every half a year since then that I'm looking into at the exact same spots. And we can see some signs of recovery happening at Glenafine for sure, and at some other sites as well.
So how do you go about standardizing the photos that you take at each site?
We have little pins down in the reef, so we make sure we go back to this exact same spot with a little quadrat that we have, and then we put the quadrat down onto those pins, and then that way we can see changes happening on the spot, so in the same location, which is quite nice. So I can overlap the images and see, there was a sponge there this year, it's not there the next year, or there wasn't a sponge, and then I can see, there's one the next year.
Presumably some of that data has been analysed already to kind of look at where recovery is happening.
Yeah, some of that data has been analysed already. Also the early years of taking those photographs, so between 2018 and 2021 roughly, has been looked at by another PhD student that was in the lab, and now I've taken over that work. And with my work I have been looking at all the years at Glenafine so far and all the years at Whirlpool as well. Glenafine is the more diverse in recovery and stabilization as well compared to Whirlpool, but Whirlpool is also definitely changing towards a more spongy environment again.
Right.
Yeah.
So you're building on the work that he had done previously.
Yes, I'm building on that work. And with my PhD, we now have the chance, because we will have 8 years of data in the scope of my PhD, we have the chance to actually get a full recovery period, hopefully. And that will allow us to look for patterns in the recovery as well, which is one part of my project that I'm doing as well, trying to find patterns of succession and recovery in those kinds of sites.
And when you're saying community dynamics, is it helpful to think of this as kind of a regenerating forest? So there's certain sponges that will show up first, and then another group of sponges, and then as it goes on, you get the next group of sponges. Is that the kind of way we should think about it?
That is pretty much the way I'm trying to figure out if we should think about that. That's exactly what I'm going to do in my succession chapter. I'm going to look at if we find early colonizers on a reef that has just been wiped off sponges, basically, and then who comes in next. It's not only sponges as well. I'm looking at the whole benthic community, or the whole sessile benthic community.
And that's the things that live on the seafloor.
Yeah,
that
the
don't
things that
move from
live,
the
that don't
seafloor.
move, yeah. So everything that grows on the seafloor. Maybe we can find some patterns of, early colonizers coming in, then other sponges or other species taking over, and then in a later stage having a plateau of certain species being there in percent abundance, basically. I also do work out in New Zealand, so my PhD is not only focused on Lough Hyne, but with this time series that we have here, we get a very unique chance to look into the community dynamics and recovery dynamics of those temperate mesophotic sponge reefs and look at it firsthand, because the reefs that we find here are in diveable depth, which we don't have that often anywhere else. We have some in New Zealand around the Kapiti Coast and Mana Island. There's some sponge reefs that are in dive depths, but the environment of Lough Hyne, which is quite calm, gives us the chance to go down to these reefs and see them for ourselves.
The sponges here are not the same species as those in New Zealand, but some are quite similar and likely play similar roles in the ecosystem. For the New Zealand part of Kea's work, she'll be looking at how sponge communities differ depending on how steep the reef or cliff face is. Basically asking whether some sponges like to live life on the edge more than others. She hopes at a number of different sites, but that depends on the opportunities. Something that keeps coming up when I chat to the team is how the conditions in Lough Hyne enable them to dive pretty much when and where they want, unlike other sites.
I love the diving here at Lough Hyne, especially compared to Wellington. It is so easy to get out and go diving because it's very calm conditions usually. And also, I don't know if it's a thing of this year or if it's a thing in general, but the visibility has been really quite nice. So we can actually see stuff and we can see everything that is happening around us, which I'm not really used to. I did a lot of diving in lakes in Germany, where at the bottom you have very, very fine sediment, and once you whirl that up, you can't see anything anymore. So I've been really enjoying the sides of the reefs underwater and seeing all the organisms interact with each other.
And obviously all of you guys are kind of into sponges and that's your main focus, but are there other interesting things that you've come across on the dives down there?
Yeah, we came across one of the anglerfish that apparently hasn't been seen here for a while, so that's really, really cool. And just today we saw a little shark, I believe. Not quite sure, we need to look it up, but that was the dive Gabi and I were just on, and we just came up and didn't have time to look at it, but I took a little video of it, and then We keep finding a lot of lobsters down there as well that are very curious about what we do, especially if we're staying in one place because we're doing work in one spot for a little while. They tend to come around and just check us out, and some of them might be have their guard up, but it's been quite fun to see things living and thriving down there.
This has long been the appeal of Lough Hyne. This tiny seawater basin, with a dense concentration of different habitats, is protected from the wild Atlantic weather. With its rich history of scientific exploration, over 1,800 species have been identified in the loch. And as they got better equipment and explored further, scientists realised that we find these same critters in deeper places in the ocean. So Lough Hyne is somewhere where you can investigate these generally harder to reach ecosystems with just a short boat trip and a quick dive. While the analysis to date of the carefully collected pictures shows sponge recovery at some sites, at others it's been quite slow. Which brings the team to question 3. Can they help the sponges return? I find James and Gabi on the jetty that's a short walk down from the lab. They've hung a cage from the bottom of it, and inside I can see some grey hexagonal tiles with a bit of yellow sponge tied on top of them.
We went out a couple of weeks ago and we cut very small pieces of sponge off donor sponges over at Whirlpool Cliff, and we've been growing them on really small tiles for the last 2 weeks. And our plan is, now they've kind of healed up and mostly sealed over the bit where we cut them, we're going to plant those back out to the sites where there aren't any sponges. We're only doing a few this year just to basically make sure the conditions are still good for those sponges, and then if it's successful this year, then next year we'll plant out a whole load of them, so we'll make a little mini population of them.
And when you say you've cut a bit off, sponges can clone themselves, so these should technically just keep on growing?
They should, yeah. So you can chop a sponge up into 50 different bits, and each one will make a new individual sponge.
So you've cut a little bit off.
Yep.
And it's probably like diameter about $2 coin, and
yeah, it's about $2 coin size. Yep, and that's normal. They grow quite slowly. So, but over time, the idea is you can't see, what you can see is a big tile that they're sat on, but underneath that, they're actually been grown onto a really small tile. These are common or garden bathroom tiles purchased from New Zealand and brought all the way especially to Ireland. And the idea is that we will epoxy those small tiles with a very tiny bit of underwater glue onto the reef. Eventually, the hope is that the sponge will grow over the tile and onto the reef area, and then eventually, 3, 4, 5 years time, we'll be able to, if we want to, cut out the bit in the centre of the sponge, remove the small tile, and then the sponge's tissue will grow back over that little hole.
Right, okay.
Yeah, so we won't leave anything behind. That's the ultimate hope.
And this is the first time trying it out, kind of?
This is the first time trying it out, yeah. So this is, so it's all a bit sort of trial and error at the moment, but I think we're getting there. I think we've got a good solution.
And this particular species is?
This is Cleona salata, quite a common species over at Whirlpool Cliff. It forms, some of the individuals over there, some of the sponges over there are maybe a metre long, really long-lived. There's one particular sponge that I remember from when I was an undergraduate student, and it's still the same size and shape as it was, which is 20 or 30 years ago.
Wow!
And my supervisor saw the same sponge when he was at Lough Hyne as a young scientist too. So it's been the same, sitting there, sucking in water, spitting it out for at least 60 or 70 years.
One more cold dive later, the sponges are in place and we're headed back to the lab.
So all went well, James?
Yeah, it did. It took a bit longer than expected, but we transplanted out 6 little sponges on the little tiles. They're now safely cemented to the reef. There was quite a large spider crab that was sat there next to me the whole dive, looking like he was ready to just pluck them off as soon as I disappeared, but I shooed him away and hopefully he won't come back and pick them off and stick them to his shell or something really cool, like in wine.
And you used epoxy to glue these ones onto?
Yeah, so we use like an aquarium epoxy, so it's non-toxic, but it's quite hard work. We mix it up underwater, so it's like all my fingers are aching now from like kneading it and making it as well mixed as possible. And a little small bit of that on the back of the tiles and onto the reef, and then just put them on. And hopefully it'll set in the next probably 4 or 5 hours, and then they'll be firm and the sponges will start to grow around them and over them. Hopefully, that's the plan anyway.
And would the spider crab eat them, or just use them as decoration?
Probably just decorations, I reckon. They look pretty cool. I guess a little tiny yellow sponge. Who wouldn't want a little tiny yellow sponge on their back?
James will return in November, and he'll check then, in even colder temperatures, to see how these sponges are doing. They're still hoping to get to the bottom as to why the sponges disappeared. But in the meantime, these sponge-clad tiles are just one of the trials underway to try speed up their return. They've also moved some rocks that have sponges on them from one area to another, and they've put down some blank tiles in areas where sponges are recovering, hoping that sponges grow on them naturally and then they can easily move them elsewhere. I ask whether what they learn will help us in New Zealand.
The techniques that we develop here at the Lough will be useful to anybody doing restoration of sponges anywhere in the world. I guess the hope is in New Zealand that we never have to use these techniques and the approaches that we're working on developing here. But they certainly should be applicable to any temperate system and potentially tropical systems too. There are quite a few places in the world where sponges have declined as a result of marine heat waves and those kind of things. And these tools could be used to help restore those populations.
But for Professor James Bell, another key benefit of the work here in Lough Hyne is the experience that these Victoria University of Wellington students have gained across the years.
This time there are 2 PhD students, and over time I guess maybe 4 or 5 PhD students have contributed to different parts of this project. So it's great for those students. They get a feel for, I guess, what environments are like in different parts of the world. The really cool thing about this project as well is it's a real life applied science question. A lot of things we do are, I guess, academic in nature, but there's a kind of real world outcome to try and understand what's happened at a marine reserve and then try and support it not happening again.
Thanks to Professor James Bell, Gabi Wood and Kea Witting from Te Herenawaka, Victoria University of Wellington. Thanks also to Lough Hyne and Professor Rob McCallan from University College Cork. This episode was produced by me, Claire Kincannon, and sound engineering was by Steve Burridge. And for the final time, for now at least, production help was from Ellen Rikers. Ellen has worked part-time on Our Changing World for the last 3 years. She's made some beautiful episodes across that time, from exploring the lava caves under Auckland City, to a story about kākahi conservation, to explaining how the Tongan volcanic eruption caused a worldwide tsunami. And as a listener, that's what you'll have heard. What you might not be aware of is every time I say production help was from Ellen Rikers, I mean all of the things she has done in the background that week to help get the show out. So, thank you for your mahi, Ellen, and best of luck with your next adventure. Ganairi and Boherlat. If you have any feedback or questions, or want to share which of Ellen's episode was your favourite, you can email ourchangingworld@rnz.co.nz. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.